Hi everybody, welcome to my channel, or welcome back to my channel. Today I'm going to do a video on microbiology. I had some positive feedback after I mentioned I might do this video, and it's also cold and flu season, and I wanted to mostly focus on bacteria and viruses and the difference between the two, considering that we are in the season of illness. So let's dive right in, okay? If you have not subscribed to my channel, please hit the subscribe button, and please also leave comments or likes. All right, so I'm gonna look at this tree of life with you all. This is where I like to start typically when I do any video on living organisms. This is like a really rudimentary book on microbes, right? So we have three domains: eukarya, which constitutes the eukaryotic living organisms, bacteria, which are prokaryotic, single-celled, and archaea, which are very similar to bacteria. In fact, scientists used to think that they were bacteria. They are not, and I will touch on this domain briefly, but we're mostly focusing on this one. You might notice that viruses are not on this tree of life. I will get to that when I cover viruses after I cover bacteria. They are, in fact, not on the tree of life. So let's look at what prokaryotic means. Because there are single-celled organisms in domain eukarya. In underkingdom Protista and underkingdom fungi, there are single-celled microbes, but eukaryotic, and most eukaryotic cells, our living organisms, are multicellular. And we're focusing on the prokaryotic single-celled bacteria, okay? So prokaryotic cells are smaller than eukaryotic cells. They also do not have a nucleus, so that's really important to specify that they do not have a nucleus. And they don't have as many specialized organelles as eukaryotes do, okay? They do have a nucleoid, but it is not surrounded by a membrane, therefore it is not technically a nucleus, all right? So let's look at what prokaryotic bacterial cells do have. Okay. They range in size from 0.2 to 10 micrometers. They're really, really, really tiny. Okay. Here we go. Into the part. Let's see. Guess we can start here. Structure of bacteria, shapes and sizes. Their shapes include spherical, rod-shaped, or spiral. They may or may not have flagella. I'm actually going to flip to a page in this book to show you some really beautiful pictures of bacteria. Now this one is not specializing in microbes necessarily, just microlife. So in this book they also are looking at like insects and other very minuscule, it's a microlife, not microbiology. Okay, so there is a distinction to be made there, right? So we're gonna look at page 29 where we can see bacterial cells. Here we go. Okay, so we have spheres. Let me slide this over for you all. We have spheres. We have rods. This is what the rods look like, almost like a little pill. And the spheres are round. And then we have other complex shapes here. filaments, rod-shaped, called elongates, where it's just like a stretched out one, and spirals. But I thought these pictures were really, really cool. Okay. And we're going to take a brief pause there before we actually focus on any specific bacteria. I'm going to go once again to this book, and we're going to talk about what bacterial cells are made of, because there's an important distinction amongst bacteria. Okay, so this is a close-up of bacteria right here. So their cell wall. So bacteria have cell walls made of peptidoglycan, but they can vary in their level of peptidoglycan. So peptidoglycan is a polysaccharide of sugar, consisting of sugars and amino acids, and it forms a mesh-like peptidoglycan layer outside of the plasma membrane that is the inside of the bacteria. Okay, so we have gram-positive and gram-negative, and the variation in bacterial cell walls affects how we treat dangerous bacteria that can cause infections and disease. Let's see, I think there's more information back here somewhere. An important distinction. I don't, maybe it's on page 32 here. Let's look. This book, you guys, I'm totally just winging this. So if I'm bouncing back and forth between books, it's because one of the books had information that I liked on one subject, etc., etc. Here we go. So gram-negative and gram-positive. Okay, so a gram-negative bacteria, it means that the cell wall has a thinner layer of peptidoglycan around it with a outer membrane. So you have your inner membrane, okay? And then you have your peptidoglycan layer that's thin, and then you have an outer membrane. Okay, then you have your gram-positive bacteria, which is just the inner membrane and then a thicker wall of peptidoglycan. This distinction is important because gram-negative bacteria are actually really difficult to kill with antibiotics. This outer membrane is hard to enter, and the thinner peptidoglycan makes the antibiotics have less of an area to target anyway. Now, some examples of gram-negative bacteria would include E. coli, which there is a picture of right here. Okay. This is also an anaerobic bacteria, meaning it thrives without oxygen. Okay. And we'll get to that briefly also. Some examples of gram-positive bacteria would include MRSA, staph, and strep. Okay. I don't know if any of you have heard of MRSA, but it is so difficult to treat because a lot of these gram-positive bacteria are actually developing antibiotic resistance because of overuse of antibiotics in our food. Like we feed antibiotics to livestock to keep them healthy, and we consume a lot of antibiotics as a species. So the bacteria that antibiotics once treated really, really well have actually started mutating and developing resistance, which is really scary. Okay, now let's get back to the inside of a bacteria. All right, the inside of the bacteria. I don't know if I have any like really good pictures of what the inside looks like, but I will say that the cytoplasm inside of bacteria contains enzymes that help in digestion, storage bodies called inclusions, nutrients, RNA, and DNA. Now, their DNA is not contained in a nucleus like ours is, and they actually have like smaller bits of DNA called plasmids, and That's important to note because sometimes bacteria can actually transfer their little bits of DNA information to each other through a process called conjunction, and they otherwise reproduce asexually. Their nucleus holds a single circular chromosome. It's a circle with several million base pairs of genes. So let's see if I can find information on the genome. Bacterial genome, it's right here. So the first bacterial genome was sequenced in 1995. Scientists still sequence bacterial genomes to gain insight into them. It's believed that some of their organelles, such as mitochondria, which help with energy, and chloroplasts, began as an actual bacteria that then kind of became part of another bacteria. So I'm sorry if I keep bumping the table. So as I mentioned, conjugation, transduction, and transformation is one way that bacteria can transfer DNA information to each other. And otherwise, they reproduce asexually. They don't have special reproductive structures. They actually just mostly reproduce through binary fission, which means that the DNA replicates, it grows in size and splits in two, or through a process called budding, where a bud breaks off and forms a new organism, or fragmentation. Each bacterial cell becomes fragmented, and then each fragment becomes a new cell. So most bacteria reproduce through binary fission, although cyanobacteria, which photosynthesizes, can use budding and fragmentation. However, some bacteria use pili to connect and exchange bits of DNA in a process called conjugation. So pili are like the little things on the outside of the bacteria. They kind of look like little arms that help them swim around. All right, so another cool thing to talk about when it comes to bacteria are anaerobic or aerobic bacteria. Okay, so anaerobic means that the bacteria actually functions without the presence of oxygen. So if you saw my video on hibernation, you saw that to produce energy, living cells need oxygen for their Krebs cycle to produce ATP. So some bacteria have developed this really cool technique of, instead of using oxygen, they use sulfur or nitrogen, which as far as like how many electrons are in the outer shell, they're very similar to oxygen. So let's look back here at look at E. coli again, right? So E. coli is a bacteria that lives in your gut, and it's anaerobic, meaning there is no oxygen available to it, which is actually really, really cool. Very, very dangerous bacteria here, though. So just, that's why we have to be very careful around feces. But it benefits us to have this bacteria in our intestines, right? So it actually is like somewhat symbiotic for us, and that's pretty cool. Another anaerobic bacteria are the bacteria that live at hydrothermal vents deep in the ocean where there's no oxygen, and they actually, where those bacteria are living in worms and helping produce byproducts that the worm thrives off of, and then there's like this whole ecosystem down deep in the depths of the ocean where you wouldn't expect to find one. Or in marshes where the soil is so waterlogged, there's no oxygen, you smell that sulfur smell, it's actually the sign of a healthy marsh. There are anaerobic bacteria doing cool things to help keep that ecosystem afloat. And we're going to look at, I'm not going to go into detail about how they absorb food or anything like that. I am going to look at that in a moment, but we are going to look at some of the uses. I'm done talking about the structures. Let's move on to the uses of bacteria, and then I will touch briefly on Archaea. That's an Archaea right there. Bacteria. So bacteria is everywhere in the soil, the oceans, plants, animals, us. They are Earth's oldest life forms and thrive in extreme conditions. The oldest fossils are like 3.5 billion years old, and they're that of bacteria. Let's see, it's actually believed that bacteria helped to oxygenate a oxygen-barren planet, so that's really, really cool. Right, so we're gonna look at good bacteria, okay? So good bacteria are basically bacteria that do things that benefit us, including cleaning wastewater. Anaerobic bacteria helps to clean wastewater, feasting on waste or oil. They are nutrient recyclers. They help break down dead organisms, releasing nutrients to be recycled. One critical nutrient that they release is nitrogen, which plants. So that's really cool. They also help with fermentation. They're used to make dairy, cheese, butter, wine, yogurt, kombucha, that's your thing. Bread requires bacteria as well as yeast. So we actually use bacteria to help us with producing food. And then antibiotics themselves come from particular bacteria through a fermentation process. Except for penicillin, there are some fungi that create antibiotics, and that's what this picture was here. That I showed you. Oh, I went way too far back. Now, if you are interested in fungi or protists, let me know, because I could do another microbiology part 2 video to go into more detail, or if you want to know more about the inside of a bacteria structure. I thought I had noted in here. Oh, here it is. Oh, I'm so sorry. Okay, so here's the inside of a bacteria. Okay, you see all microscopic image of the bacterium. Shows their cell wall, cell membrane, nucleolus is pink, and then the orange and blue are their organelles. Organelles. Okay. So here's penicillin. Penicillin is grown from fungi and is used to treat. It was one of the first, if not the first, antibiotic, and helps fight off some pretty cool serious infections. So lots of good bacteria that help decompose, live in our guts. I don't know if you all take probiotics or eat yogurt, but the fermentation process in yogurt creates probiotics that are beneficial for digestion. We have other symbiotic bacteria in our guts and our whole body, actually, technically. And I I discussed the hydrothermal vents and salt marshes, nitrogen fixation. So most bacteria are actually like benign, with some of them being symbiotic or beneficial, and just a small percentage being really, really dangerous. Now, here's the thing about bacteria. If you have a bacterial infection, such as MRSA, strep, staph, I think technically gonorrhea is also a bacterial infection. Like an ear infection, that's just bacteria. That's like a secondary infection from a virus, which I'm about to get into here shortly. And so you can take antibiotics for those things to help fight and kill the bacteria. Okay, my nails were not even on screen just now. I apologize for that. So mostly when you have a bacterial infection, it's different than a viral infection, okay? Which is why if you don't know the difference and you have a viral infection, and it's important to know the difference, which your doctor knows the difference. Okay. Also, antibiotic resistance is a reason why most doctors nowadays are more hesitant to prescribe antibiotics. So keep that in mind if your doctor ever acts like not super amenable to prescribing antibiotics. Now, we're going to briefly touch on Archaea. Archaea is the other domain of prokaryotes. Now, I will say, if you look at kingdoms, there are actually, depending on who you talk to, 5 or 6 kingdoms. We're talking about domains. So some scientists include Archaea as a domain in a kingdom, and same with eubacteria. And then the eukaryotes have plants, animals, protists, and fungi. So they have four kingdoms within their domain. Whereas the domain of Archaea is just, it's also a kingdom Archaea. Okay. And it's different. They're different from bacteria because they have a different type of cell wall. And we're going to look at that, page 49. They were discovered after bacteria, and they were thought to be similar to bacteria, or a type of bacteria. So in many ways, Archaea resemble bacteria. That's why they were first considered weird bacteria. A membrane and cell wall surround most Archaea, and capsule encloses everything. The interior holds cytoplasm with ribosomes, a nucleoid, plasmids, and storage bodies. What makes them different? Archaea have some distinctive shapes. They can be shaped really thin, almost rectangles, triangles. And chemically, their cell walls lack peptidoglycan. Now, peptidoglycan is what we talked about, referring to gram-negative, gram-positive bacteria. They also have different chemicals in their membranes and their flagella, and their storage bodies hold different material. Their ribosomes act more like those of humans than of bacteria, and they differ genetically. So they did have their genome sequenced in 1996, and they have many genes not found in bacteria or eukaryotes. They have inactive DNA segments called introns. The reproduction is similar to that in bacteria through binary fission, budding, fragmentation. It's important to note too that a lot of the extremophiles, or organisms that live in really extreme environments, belong to this domain. So they can live in like boiling temperatures, some of the hottest places, acidophiles, high acid conditions, cool ocean waters. So they actually have extremophiles that live in places you would not expect to find them, which is really kind of cool. Okay, so that's all I'm gonna say about Archaea. And I'm gonna move on to viruses. Viruses, invisible invaders. So, viruses are not living organisms, okay? They are literally just genetic material, RNA or DNA enclosed in a protein coat. I don't have a picture of that. Thought maybe I did. I do not see any pictures of viruses. I apologize. I will tell you that if you Google it, you could find a picture of your quintessential virus, which looks sometimes like it's shaped like this with a little thing to insert the protein capsule into your cell. So what a virus does is it hijacks your cell. Hijacks your cell, hijacks your cell, and uses your cellular machinery to reproduce. Now, that's important because to kill a virus, oftentimes you have to kill your own cell. A lot of viruses your body can fight off on its own, the common cold, unless you get a secondary bacterial infection that you need antibiotics for. The flu is a virus. A lot of times your body can fight it off on its own. So sometimes if you have a virus, your doctor will say you just have to let it work its course in your body. But there are some viruses that are not so inoculate. They are very, they stick around. And you probably have heard of HIV. There are other STDs that are viruses, chickenpox, polio. So how do we combat a thing that is not living and it just takes over your body, right? The capsids that protect the genetic material of the virus also, there's also a protective envelope of proteins, fats, and carbs that help the virus attach to a host. They cannot reproduce on their own. They have to attach to a host, take over their cellular machinery, and use your cells to reproduce. That means it's not a living thing. Most scientists agree that they're not a living thing because they cannot reproduce on their own. Right. So how do you treat something like that? Well, we have vaccines for many viruses: polio, chickenpox, shingles, which is basically chickenpox. Let's see, herpes. I don't know if there is actually a herpes vaccine. I think they have one. Flu vaccine, COVID vaccine. COVID is a SARS virus. So that's one way that scientists have been able to help fight off the viruses, is to have a vaccine available for it, so your body, your immune system is strong. It knows what to do when it encounters that virus and it fights it off effectively. And there are also antiviral medicines for viruses that they don't have vaccines for. So let's look at how you do that. You can block receptors so viruses can't bind to and enter your cell. So you block the receptors that the virus would use to land on your cell and insert into your cell, and it could block that interaction. Some antivirals boost your immune system or lower the viral load, or the amount of active virus in the body, and sometimes you do have to actually kill off some of your own cells to do that. There's antiretroviral that blocks the different stages of the virus. This is used in HIV. It's difficult to cure HIV because once HIV infects a person's body, it integrates into the host's genome of several cell types. So the virus takes over the cells and actually inserts into the genome. Then they hide in lymphoid tissue, lymph nodes, liver, and spleen. It's really, really tricky to target those. So an antiretroviral blocks different stages of the virus's life to give the immune system a chance to fight it off. But even if you fight most of it off, you will always still have some lying dormant. So it's one of those viruses, once you have it kind of like for life, right? And there are other viruses out there like that. It's rare, but they're out there. And so the important distinction between viruses and bacteria is that viruses are much, much harder to treat because there's less to target without targeting your own cells. So there you go. That is the basic gist of the video for today. And it's very rudimentary microbiology. I didn't have great diagrams. I was really just working with what I had here, but I wanted to do this in a time where we are experiencing high levels of viral infections and cold and flu season. And so the hand, foot, and mouth, that's a virus. My child had that over the summer. And yeah, they're pretty scary, but in general, mostly your immune system can fight those things off, right? So anyway, if you have any questions or you want like a part 2, let me know. I would be happy to film a part 2 of this video at some point. If you absolutely hated this video, please tell me why, other than my lack of organization and pictures. And otherwise, I hope that everyone has a fantastic day. Thank you.